Welcome to Molecular Nutrition, where food becomes molecules, molecules power cells, and cells sustain life. Today we shall continue with proteins and amino acids. So module 3 we shall be dealing with proteins and amino acids. As we know, amino acids are the building blocks of proteins, and in this module we're going to be learning a lot about those building blocks. One key thing about this module is that we shall be considering proteins and looking at them as functional molecules in nutrition. And when we say functional molecules, we're looking at amino acids as the foundation of proteins, and of course the role proteins play in metabolism and the roles they play in growth, in immunity, and in health generally. But amino acids are functional molecules in the sense that, well, the basis for they form the basis for proteins. Okay, there's a lot of chemistry involved, and we're going to be looking at the chemistry of amino acids and how they form the building blocks for proteins. And I would like to warn you here, more like encourage you. So we're going to be looking at lots of structures, but the reason we are looking at those structures is not because we want you to cram them. It's because they aid in the understanding of proteins. You can't understand how proteins work without understanding their building blocks, and the building blocks of proteins are amino acids. And of course, you can't understand those amino acids if you don't understand their structures. So we'll just be looking at them but the whole emphasis is that they help you to understand what is going on. So it's good to understand the roles that proteins play in human nutrition. And the first thing that comes to your mind when you think about proteins is enzymes. So enzymes are proteins, and of course enzymes catalyze metabolic reactions. So think about the hundreds of metabolic reactions that we have in our cells, hundreds of thousands literally of different metabolic reactions that we have in our cells, and think about them being powered by enzymes. Okay, enzymes, apart from that, proteins also are involved in structural components of tissues. They form the structural component of tissues. What comes to your mind when you think about proteins forming the structural component of tissues? What comes to your mind looking at all the things that we have done in a particular tissue? Does anything come to your mind that when you think about proteins? In my own mind, I'm thinking about different components of the blood. You know, the blood is connective tissue, and there are many components in our blood. When I think of hemoglobin is a protein that is responsible for the transport of oxygen in our blood. When I think of myoglobin in muscle tissues, they are also proteins, and we'll be looking at in a bit the kind of proteins that they are. But let's leave it as that. When you think about structural components, what other things come to your mind? When maybe this time not in tissues, but when I think about maybe tissues, When I think about the skin as a tissue, what do you think of? Our skin is largely protein. And what is that protein called? Our hair is largely protein. And what is that protein called? When you think about keratin, I'm sure you've heard that protein before. These are the proteins that you find in the human hair and skin, and also nails as well. Okay, so in that sense, proteins play a structural role, but we also see, in fact, at the cellular level, we see a very important aspect of proteins: transporters, receptors. So we've been looking at all the transporters, we've been looking at different receptors, we've been looking at ion channels. So we can imagine all those things that we have done. They are largely proteins. They are largely proteins. If you also think about hormones, immune bodies or immune molecules, all these are proteins. Okay? So it's good that you already have that idea that all those things are proteins. Okay, so moving to the next slide, we're indeed going to be looking at the molecular structure of different amino acids. Our amino acids are the foundations of proteins. And we're going to be looking at this with regards to metabolism. Okay, we're going to be looking at it with regards to nutrient utilization. What can you think of around that? We're going to be looking at how, the structure and chemistry of amino acids and proteins that forms the basis of growth and tissue repair. And of course, all this will be in preparation for later courses when we'll be looking at the direct application of proteins in diets and the different types of diets at different life stages, the composition of proteins in those diets, and how we can kind of like capitalize or work around our knowledge of those proteins in managing different diets, especially for different disease states. But that's going to be at a much advanced stage. For now, what we're going to do is just to learn the foundations of these proteins. So what are the learning objectives? So the first learning objective is to understand the chemical structure of proteins, the chemical structure of amino acids, and the classification of amino acids. In understanding the chemical structure, well, we'll have to really understand the first 20 amino acids, right? And this is important. So we're going to be going through the details of the first 20 amino acids because they form the building blocks of proteins. So it's important also that we memorize them. So we'll have to know them. We have to know what makes them what they are. So if I'm going to give an analogy in an analogous manner, understanding the first 20 amino acids, as we call them, is one of the objectives of this course. So it will require that we memorize them. And the same way when we were in primary school, we had to memorize the multiplication table. And it was based on that memory that we began to understand other concepts of multiples of different numbers. So in the same way, to understand proteins, we need to definitely have a basic knowledge of all the first 20 amino acids. So it's very important. So I'll recommend that you take your time in really learning them. It's sometimes very difficult to memorize new things, but it's not impossible. So you have to disconnect from all maybe different sources of distraction. You have to try to make your attention span a bit longer, and you would more importantly need to focus and consider them every day. I promise you, if you look at them every day, in less than 3 days you will master all the amino acids and all their different categories and all their different subcategories. Okay, so it's precisely after mastering that you will then be able to master other things and learn the differences, between the essential amino acids and the non-essential amino acids. Okay, so this learning all the different amino acids is actually dependent on you having mastered the first 20 amino acids. We'll be looking at formation of peptide bonds and in a protein structure, and condensation reactions and role of hydrolysis reactions in the forming of peptide bonds. And we'll also be looking at the functions of proteins in biological systems. Then, more importantly, one of the learning objectives of this course is to relate the protein structure to nutritional and physiological functions. Okay, so in this slide we consider the nature and the basic structure of amino acids. So the basic structure of amino acids is, first of all, a central carbon. An amino acid has the central or alpha carbon, and if you look at this example, we can see here this carbon is the alpha carbon. Okay, all amino acids have that. Then connected to this is the amino group, and we can see the amino group here, right? So you can see here the amino group. Connected to the amino group is the carboxyl group, and we can see right here the carboxyl group is the COOH group, as it were. So the C double bond OH. So it's the same way that you write it, if you want to write it like this, right? And that carboxyl group is connected to the alpha carbon. Next, as the basic structure of amino group is the hydrogen atom. So we can see clearly here the hydrogen atom. And one last structure of the amino acid is the R group. The R group. And this R group is usually what varies in all amino acids. So at this point, we will need to learn the first 20 amino acids. So we're going to be learning them, and like I said, we need to memorize them. So, but the first thing, we can go through them one by one. Then I would show you the different ways of learning them easily. So we have glycine, we have alanine, we have valine, we have proline, we have serine, right? We have asparagine, we have phenyl, alanine, we have tyrosine, histidine, number 9. We have cysteine, threonine, methionine, leucine, tryptophan, glutamine, aspartate. It's also called aspartic acid, and it's acidic. It's one of the acidic amino acids. We have isoleucine. We have glutamate. This is also another acidic amino acid. It's also called glutamic acid. And we have arginine, and of course, last but not the least, lysine. So these are the first 20 amino acids, and it's good. So you're going to be hearing their names in different contexts again and again and again. Indeed, there are different things that we'll need to learn about them, and I'll be using this slide to demonstrate the things that we need to learn. Okay, so here in this slide, we're looking at the different amino acids. So we begin with non-polar amino acids. So why do we call them non-polar? We've talked about polarity previously, and it means that they don't have a central charge. They have a net charge of 0 in that sense. And you can see how their net charge is 0, because if you look at the R group, and in this case, the R group is the group that has been highlighted here. So you can see the R group in all these cases highlighted. So if you look at the nonpolar amino acids, you notice that the R group they have no charge. If you look at alanine here, the R group has no charge, net charge of 0. So in that case, the delta positive and the delta negative cancels out each other, so the net charge then becomes 0. So this is why we call these amino acids non-polar. Okay, so we then go straight into which are those amino acids that are non-polar, and the important thing is learning first the names of those amino acids. There's an abbreviation that is usually used, and that abbreviation is GAVLIMP. Okay, and for what does this stand for? So we have G for glycine, we have A for alanine, we have V for valine, we have L for leucine, we have M for methionine, we have P for proline, and then we have F for phenylalanin, and W for tryptophan. So usually we normally call it GAVLIMP FW. GAVLIMP FW. Okay, so again we've missed out isoleucine. So you see the isoleucine is the I in the GAV limb. So I don't know if this helps you. If you already have an acronym, that's fine, but you can also memorize GAVLIMP FW to as a mnemonic or memory aid to learn the nonpolar amino acids. So at this point, I would like to show you the single letter form of amino acids, and of course the 3-letter forms of amino acids. So we can see here that this amino acid, for example, G, glycine is G, but it has a 3-letter of GLY. Alanine is A, and it has a 3-letter of ALA. Valine, V, 3-letter of VAL, Val. Leucine is L, 3-letter of LEU. So in most cases you can see it's the first 3 letters. Isoleucine is an exception, is I, 3-letter of ILE. Methionine, M, 3-letter of MET. Phenylalanine is an exception. So phenylalanine in this case is F. So we can see that it's an exception. So there are some exceptions to those amino acids where their first letter is different from the actual letter of the amino acids. So we'll be learning them in a bit, but I will just advise that you spend some time with them so that you actually memorize them. The same thing for tryptophan. Tryptophan is W, and I guess it has to do with the benzene ring, but the 3-letter is TRP. Proline is a P, and of course the 3-letter is PRO. But these GAVLIMP FW form the nonpolar amino acids. In understanding the next, talking about breaking the amino acids into different, based on their characteristics and properties, we then look at acidic and basic amino acids. So there are just 2 acidic amino acids, and they are aspartate, like we considered, and aspartate has the letter D, and glutamate has the letter E. And of course, if we look at the first 3 amino acids, ASP, GLU. So aspartate and glutamate, they form the acidic amino acids. Going to the basic amino acids, we have lysine, we have arginine, and we have histidine. Lysine, arginine, and histidine. So lysine is a K, it's also inconsistent. Arginine is an R, it's also inconsistent. Histidine, H. But of course, the first 3-letter for lysine is LYS, ARG, HIS. So it may sound a bit confusing now, but you will get used to it, and just take your time to learn the first letter and the 3-letter of the amino acids. So we'll move to aromatic amino acids. So the aromatic amino acids are the amino acids that carry a benzene ring. So you can see them, a benzene ring. And so you have phenylalanine as one of them. And phenylalanine, remember, is an F, first letter, and 3-letter PHE. Tryptophan is also an aromatic amino acid. And do you remember the first letter for tryptophan? W, and the 3-letter TRP. Okay? Tyrosine is also an aromatic amino acid. Can you guess the first letter for tyrosine? Y, and the 3-letter TYR. Okay, so take this, learn them in beats, and get used to the first letter and the 3-letter. So we move to the other types of amino acids. There's also the hydroxyl amino acids. So the hydroxyl amino acids usually have that OH as part of the R group. Don't be confused with this structure of the amino acid. You see it's a little bit different. So it's not written. So this is where the carbons are. So this carbon, and this is carbon carrying an H here, and of course connected to the hydroxyl group. Okay, and of course this carbon is connected to the C double bond O group. So don't be confused. So if you see a different structure, don't be confused. This is how it's related to what you already know. So the hydroxyl amino acids, so the first one we're considering here is serine. And serine, the first letter for serine is S, and the 3-letter is SER. Next, we have threonine. We've come across threonine before, you remember? So the first letter for threonine is what? T, and the 3-letter is THR. The same, we came across tyrosine. Tyrosine, the first letter for tyrosine is what? Y, and the 3-letter TYR. Okay, so take it one at a time and you memorize the first letter and the 3-letter. You can also use flashcards if it works for you. Do whatever works for you. Okay, so in learning the building blocks, like I told you, it's like you're learning the multiplication tables when you were in primary school. So you just have to learn them. Take your time. Use whatever means. So I've prepared this as a kind of like a blank check to test your memory of the first 20 amino acids. And so you can use this to learn or to see or practice what you have learned. So we can do this as a small exercise. So the non-polar, you remember. So what you should do is to first write the first letter and then the 3-letter and then name the amino acid. So we can just quickly do this. Non-polar, you remember Gavlimp. You remember Gavlimp? Yeah, that's it. So Gavlimp, FW. So and you know what G stands for. So glycine, alanine, valine, leucine, isoleucine, methionine. You remember P? You remember P? Proline. You remember F? Phenylalanine. I remember W? Tryptophan. So these are the non-polar amino acids What about the carboxyl amino acids? The carboxyl amino acids are also called the acidic amino acids. And do you remember them? So just as a recall, for the acidic, we have the D and we have the E as the first letter. And what does D stand for? D stands for aspartic acid or aspartate. E stands for glutamic acid or glutamate. So don't forget DE. In the alphabet, D follows E. And of course, A, aspartate, comes before G, glutamate. You can use the same logic for the amine amino acids. So the amine amino acids are basic in nature. So they are the basic amino acids. So what are those amino acids? We have the K and R. K is lysine, R is arginine, and of course we have H, which is histidine, HIS, right? So those amino acids form the basic amino acids, the lysine, arginine, and histidine. So don't forget KRH. Next, we move to the aromatic amino acids. So what are the aromatic amino acids? The first one that you shouldn't forget is tryptophan, W. Tryptophan is very important because it's essential. It's an essential amino acid. We'll get there. Okay, so tryptophan. We also have phenylalanine. It's aromatic. You know, we considered it earlier. So you remember phenylalanine, the amino acid that sounds like F, phenylalanine. And we have tyrosine as aromatic as well. So the three-letter word for tryptophan, TRP, phenylalanine, PHE, and tyrosine, TYR. So we then move to the hydroxyl amino acids. So they are the amino acids with the OH group. We considered them earlier on, if you remember. And there are three of them. So we have the serine, we have the threonine, and we have the tyrosine as well. So they have the OH group. Serine, three-letter word is SER. Threonine is THR. And tyrosine is TYR. Next, we have the amides, the amino acids with the amide group. Do you all remember the amide group in organic chemistry? CONH2 group. So the amide group, the R group, is CONH2. So what are the amino acids that fall into that category? The first one is asparagine. So you can use the logic of the carboxyl. So the carboxyl group. So you have aspartate and glutamate. But in the amide, you have asparagine and glutamine. Asparagine is an N and glutamine is a Q. So N, three-letter for N, asparagine, three-letter for glutamine is GLN. So this is the single letter, or these are the single letters, and the three letter forms of the first 20 amino acids. So if you look at all these amino acids, you notice that we have practically covered the first 20 amino acids. So, but I think there's one or two that we have not covered, and so for example, cysteine is not here. So we have cysteine, and the letter is C. Okay. So in this slide, we shall be considering the essential amino acids. So what are essential amino acids? Essential amino acids, the amino acids that must be supplied in diet because the body cannot synthesize them. So they therefore have to be in diet, as it were, and they usually vary. So what is essential? The amino acids that are essential for different organisms vary, but we're going to be looking at in humans. So for us, what are the things that make those essential amino acids? It's interesting that apart from the amino acids that are essential, varying from organism to organism, even in humans, the essential amino acid slightly varies depending on whether we are adults or children. Okay, but the important thing is that there are 9 of these essential amino acids that have been identified in humans, and because of that, as nutritionists, you therefore know that it's important, talking about diet quality, that the sources of our protein, they have these essential amino acids. So this is one of the reasons why we have to learn the amino acids as the building blocks, because as nutritionists we always have to keep our eyes out there. So from a public health perspective, we are the guardians of the health of the public, the foods that are available in our society. Okay, so in terms of quality, how much of those essential amino acids are in our diets, we will soon see the important things that we need to keep at the back of our minds. But for now, let's dive into the building blocks. So there are 9 of those essential amino acids. So if you look at them, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Okay, so but there's an easy way that we normally memorize them, so you could learn that as well. So we normally use the acronym PVT Team Hall. So PVT Team Hall. So usually we skip that A, and it becomes PVT Team Hall, HLL, and 1, 2, 3, 4, 5, 6, 7, 8, 9. So making those 9 essential amino acids. So don't forget PVT Team Hall, as it were. So you skip the A for Hall. And yes, so it's also good to, as you're learning the acronyms, you're learning the 3-letter words for the amino acids, and you're learning the single letter words for the amino acids. So we can then rearrange them. So for P for phenylalanine, V for valine, T for tryptophan, then T for threonine, the second T for threonine, I for isoleucine, so the 3-letter word, M for methionine, H for histidine, L for leucine, and the other L for lysine. And if you count, always make sure it sums up to 9. Okay, so and that's it. So then the other thing is to learn what is the single letter for those essential amino acids. So P, V, T, Tim, Hall. Unfortunately, the single letters are not the exceptions, in the sense that they coincide with the 3-letter as well. So they are not the exceptional single letter amino acids. Okay, so that's it for that. So in this slide, we will then look at the non-essential amino acids. So what are the non-essential amino acids? The non-essential amino acids are those amino acids that can be produced or synthesized by body cells. So they don't need to be in our diets. And individuals who do not synthesize sufficient amount of these amino acids due to illness or for whatever reason will need supplements. But for most of us, most normal people, the body synthesizes these amino acids. So it's good to also learn them. But of course, if you know the 20 amino acids, you know that the balance of the 11 amino acids, they form the non-essential. Okay, so it's good that you also get the 3-letter word for each of them. You practice that: alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. Okay, so you can also add a mnemonic to that if you want, but what is important at this point is to get the 3-letter for the amino acid and of course the single letter. So ensure that you practice it until it's part and parcel of you. So what are the single letter for the 11 non-essential amino acids? At least it's good you remember. So let's practice it. Alanine. What is alanine? Alanine is an A. Arginine is an R. Asparagine, do you remember asparagine is an amide amino acid? It's an N. Aspartic acid is a D. Cysteine is C. Glutamic acid, you know the two acids, they follow themselves, D and E. That logic is helpful. Glutamine is also an amide amino acid, and it's a Q. For me, the aspartic acid and the glutamic acid, I use the logic of D and E in the alphabet. And for the amide containing amino acid, I use the logic of N and Q. So I combine them as DENQ, like thank you, that kind of thing. So D is for the two, DE is for the two acidic amino acids. That's D for aspartic acid, E for glutamic acid. And of course, I use N and Q to represent the amide containing amino acids. So N for the asparagine, and Q for glutamine. So thank you. So you then find that yes, it helps me then Q to remember, and you should also do the same, at least before you get used to it. Okay, so for glycine is a G, and proline, do you remember proline? Proline is a P, serine is an S, and tyrosine a Y. Okay. Okay, so capping it all, you may want to practice this. We've already done the nonpolar, we've done the carboxyl, we've done the amine, we've done the aromatic, we've done the hydroxyl, we've done the amide, and we've finished the non-essential. So if you're able to supply all the amino acids from all this based on their characteristics, and you're able to categorize all the amino acids in their different categories using the 3-letter word and using the single letter, you would, it's the test for how confident you are with this assignment. So I would like to give you this as an assignment, so do it until it becomes part and parcel of you. Okay, so we move to peptide bonds and the formation of peptide bonds. So what are peptides? So you remember when we looked at carbohydrates, we looked at two monomers or two monosaccharides coming together to form a disaccharide. In the same manner, in the building of proteins, amino acids come together, they bind together, and they form peptides. And whether they're dipeptides or whether they're oligopeptides, so dipeptides are 2 amino acids, and oligopeptides are 3 or more amino acids. So whether they are dipeptides or oligopeptides, they form bonds, and these bonds form the building block for proteins, like we have said. And yes, in this slide we're going to be looking at how those building blocks are formed. So you recall that while we're looking at amino acids, so if we look at amino acid 1, amino acid 2 coming together, we see here the C terminus of the amino acid and the N terminus of the amino acid. So if you look at amino acid 1, you see the C terminus, you see amino acid 2, the N terminus. So what you notice is that in a condensation reaction, water is lost. And in the losing of this water molecule, a dipeptide is formed. So you can see that this formation of a dipeptide happens as a result of the mono amino acids in a condensation reaction losing water to form this dipeptide. So the bond that is formed when two amino acids comes together is called a dipeptide bond, and this happens in a condensation reaction where water is lost. So in the same way, we can look at a hydrolysis reaction, which is like the reverse. Earlier on we talked about a condensation reaction, but if you break a dipeptide here in a hydrolysis reaction by adding water, what you form is you form two amino acids. So in a condensation reaction, you can combine two amino acids to form a dipeptide, but in a reverse hydrolysis reaction by adding water, you can break a dipeptide bond to form amino acid 1 and amino acid 2. So in this slide, we're going to be looking at how amino acids play a role in growth and health, basically. And so this is connected to, you remember that when we're talking about growth, tissue growth, children growing, they need a lot of proteins, right? And that's why in the first 1,000 days of the life of a child, it's very important that the child gets the required amount of proteins. And it's not just about the protein, it's also the type of protein. And we know that the essential proteins are very necessary at this time. And amongst them, you remember the essential protein or amino acid leucine. So leucine and many of the branched chain amino acids are very important, because when they are not available in especially a child, it could lead to muscle wasting. Now, why is this so? Because these essential amino acids are very important for protein synthesis. And of course, if they are not available, what happens when the body is synthesizing protein? I'll try to see if I can demonstrate it. So imagine you have a sequence of protein that is needed or needs to be produced, and your body is forming those proteins. Let's say, for example, in your muscle cells or wherever. Let's imagine that this is the protein, and in that protein is the sequence of amino acids. Now, if the body needs leucine, and leucine is not available, what happens is that during protein synthesis, that protein is truncated. So imagine the body needed to produce a particular protein that has this sequence. Leucine being an essential amino acid, if it's not available because either the diet is very low in that protein, or for whatever reason, what happens is that when the body in protein synthesis gets to that amino acid like leucine and it's not available, what happens is that the body stops the process. So that protein will not be produced because the amino acid needed for it to be produced is unavailable. So if this were the required protein, it will mean that the body cannot just produce it. Why? Because leucine is not available. Okay, and that's the implication, and that's why essential amino acids really need to be present in the diet. So If it's in the muscle, in this case, those proteins are not available and it will lead to wasting. So still on the amino acid leucine, this is just by way of information so that you can see the importance of essential amino acids. There is a very important signaling pathway. I'm not going to go into the details now, and that pathway is called the mTOR pathway. The mTOR pathway is the body's master growth regulator, and it's a very complex pathway, but we call it mTOR C1 pathway. And leucine is a very important amino acid for that pathway. So if leucine is unavailable in diet, what happens is that pathway cannot happen, and it leads to growth stunting. So the point here is that without the sufficient supply of these building blocks, the body cannot initiate protein synthesis in the muscle, and this is usually the basis of stunting that we have in children. So I'm sure you've heard about the phenomenon of stunting, child stunting, where children have, they have low height for their ages. So when it comes to growth, at every age, you expect a particular height for the child. So a child that is 5 years, a child that is 8 years, a child that is 10 years, you expect a particular height. So if you're having a child that is not receiving adequate diet, adequate essential amino acids in the diet, and so on and so forth, this is what usually leads to stunting. So you can then find a child that is 10 years that is still, it's short for his age in that sense, because if you repeatedly do not give a child all the essential amino acids needed in diet, this is what happens. Now, there is an amino acid that is connected to a disease called pellagra. Pellagra is a disease that happens due to the deficiency of niacin or vitamin B3, and it has its consequences is dermatitis, diarrhea, and dementia. It's a killer disease. Not so common these days, but used to be very common in the 1960s, 1970s. And the reason for this disease is due to the unavailability of tryptophan. And tryptophan, you remember, is an essential amino acid, right? It's an essential amino acid. Do you remember? Yes. So tryptophan, the 3-letter TRP, and what is the single letter for tryptophan? Do you remember? So you should ask yourself this question, W. Okay, so linking amino acids to health implications, this is how everything is connected. So when you're listening to those Milo Bonvita adverts and you're listening to all the essential amino acids in Milo, yada yada yada, yes, this is why all these things are important. So in your own case, you should be very much interested in the foods that have this essential and non-essential amino acids, and we'll come to that in a bit. What are the other implications of peptides? There is a a clinical implication for sports nutrition. Now, I don't know how much you have an experience of sports nutrition, but a very common practice in sports nutrition is to give athletes, to give them drinks or protein drinks, or it comes usually as protein drinks, and you in those protein drinks you have peptides. So you have short peptides in those protein drinks. So you don't have amino acids per se, but short peptides. And the reason why you have those short peptides is because you want a situation in which you're really efficient on energy. Why? Because the body absorbs peptides with lesser energy than single peptides or single amino acids. So if you look at the amino acids, if you look at the drinks that are usually given to athletes, you find that they have, apart from glucose, short polypeptides, and this helps in aiding the body's absorption, because if they were in just amino acids like glucose, the body is going to use a lot of energy in absorbing them. A very good example of this is what we notice when you give a high protein diet. When you administer a high protein diet, the basal metabolic rate increases, so the body burns more calories in trying to absorb those amino acids and to put them together as proteins. So the body burns energy. But in a case in which you're trying to conserve energy, in the case of athletes, you usually administer short peptides. This is it's a little detail, and it's a common practice, and usually we consider it when we're looking at sports nutrition. In this slide, we're going to be discussing protein structure. So we have 4 levels that we usually use to explain protein structure. At the first is the primary structure of the protein, the secondary structure of the protein, the tertiary structure, and the quaternary structure. So these are the 4 structural levels of proteins, and we can delve into what each of them means. So what makes up the primary structure, what makes up the secondary, what makes up the tertiary, and what makes up the quaternary? So we will be looking at that in the subsequent slides. The primary structure of a protein is just its amino acid sequence. To put it very simply, it's the amino acid sequence. And a very good example is this protein, insulin. So if you examine the protein insulin, you can see that it's just a long sequence of different essential and non-essential amino acids. So you can look at them: asparagine, tyrosine, cysteine, asparagine. So it's just a sequence of 2 long stretch of amino acids. So we call one the A chain and the other the B chain. But then on each of the chain, you notice that you have cysteine, two cysteine molecules. So you remember that cysteine, the cysteine amino acid is the group of amino acids that has an SH. We treated it last, and that SH we call it the tile group. So you remember I treated it in the last one, but I'm mentioning it now. So because of its implication for insulin. So in the insulin protein, we use this protein as a very good example of the primary structure of amino acids. Two long chain of proteins, so amino acids combine to form a protein, two long chains joined by the cysteine tile group, the SH group. So you see one tile group at this point, and you see the other tile group from two cysteine at that point. And in a nutshell, that's what you have in insulin. You remember the insulin protein that is responsible for the transport, is the hormone that is responsible for the absorption of sugar into the cells. Okay, so that hormone, which is a protein, is a very good example of the primary structure of proteins. Okay, now we consider the secondary structure of protein. So what is the secondary structure of protein? The secondary structure of protein has to do with some folding. So, as opposed to insulin, which was just a single chain, joined by two sulfur-containing amino acids, cysteine, in the secondary structure of a protein, you have a situation in which on the same unit of the protein, you have some foldings or bonds. You have some interactions between the atoms of the amino acids that form the backbone of the polypeptide, and the bonds that form as a result of that interaction leads to a particular folding. So a very good example is what we call the alpha helix. So if you look at the alpha helix, a very good way to demonstrate it is, when you have a structure that looks like a helical structure, and you have that structure, obviously you have different amino acids. So you have some interactions or bonds that form between some of those amino acids, and some of them hydrogen bonds, and it leads to the formation of a folded helix. And that's what, instead of you having a straight chain of polypeptides, because of that you have a helical structure. And that's why we use examples of the alpha helix and the beta pleated sheets as examples of protein foldings. In the beta pleated sheets, you have this kind of foldings, but it's also from different amino acids, the bonds from the different atoms in amongst the amino acids that form that polypeptide. Bonds are formed, and those bonds lead to either the helical structure or the sheet structure. Now we look at the quaternary structure of the protein. if you look at the proteins that we have been looking at, so looking at the alpha helix, looking at the beta pleated sheet, looking at insulin, we've been considering a single unit. So in the primary, it's just a single unit, two single units, with a tile group at cysteine binding together. In the secondary structure, also bonds, hydrogen bonds forming within one subunit. But in the quaternary structure, you have multiple subunits. So within one subunit, you have interactions like we've discussed above that makes the polypeptide chain fold in a particular way. But you then also have interactions between the different chains of the amino acids. So take, for example, if you look at this protein, you can see that you have 4 subunits. 1, 2, 3, 4. So you have your alpha chain subunit 1, you have your beta chain subunit, alpha chain subunit, alpha chain 1, alpha chain 2, alpha chain, beta chain 1, beta chain 2. So, but the key thing, what makes this a quaternary structure of a protein, is that within each chain there are foldings, based on the different amino acids that make up the polypeptide chain, based on the hydrogen bonds and other bonds that lead to the atoms in the amino acids forming a folding. But at the same time, you have interactions between each of the subunits or each of the chains of the polypeptide chain. And this structure is actually what represents the quaternary structure of proteins. And a very good example is hemoglobin. So hemoglobin is made up of 4 subunits. So you have the alpha chain 1, alpha chain 2, beta chain 1, beta chain 2, and somewhere you have on one of the subunits you have a place where iron binds and a place where the heme binds. So in the function of hemoglobin, you know that hemoglobin is the protein that is responsible for the transport of oxygen. So on the heme group, obviously oxygen binds, but you also have that iron group that also binds in one of the subunits, and this is how the hemoglobin functions in transporting oxygen. And when we look at this, hemoglobin being a protein, oxygen carrier, oxygen transport protein, made of 4 subunits. This is each subunit being made up of amino acid foldings. So there's an intra folding and there's inter bonds or inter bonds that connects each of the folds or each of the chains to one another. And it's this description of the intra and the inter that describes the quaternary structure of a protein. In this slide, we'll look at protein denaturation. What do we mean when we refer to a protein being denatured? So, and what are the implications of protein denaturation? So, in a nutshell, we refer to a denatured protein as a protein that has lost its native structure. And what can make a protein lose its native structure? If you heat that protein, if you expose that protein to adverse pH changes, or if you apply some chemicals. So a protein that is exposed to either all these insults, what happens? It becomes denatured. And a lot of things happen when a protein is denatured, and we'll be looking at practical examples of protein denaturing. When a protein denatures, it's not necessarily a bad thing, but one thing that happens is that a denatured protein will lead to loss of structure and loss of function. So biologically, it could lead to the protein being inactive. But we use denaturing in the food industry. So denaturing is used in the food industry for a lot of positive things. So denaturing is used to break down proteins into their smaller polypeptides. I'll be looking at examples of this. So first of all, what happens in a pH denaturing? So let's consider pH denaturing. So if you change the pH of a protein, or you heat it, or you expose it to chemicals, what happens? That protein can lose its 3-dimension structure, and it then goes back to its unstructured proteins, but you see it physically. An example is casein, which is a protein that is found in milk. So have you seen coagulation of milk? Have you tried putting your 3 crowns milk or your peak milk, or either in the powdered form or in the evaporated form? Have you tried putting orange or lime into your milk? What happens? It will form coagulants. So you will see like precipitates, small colloids of coagulants emerging from that milk. What happened was when you put lime has a very low acidic pH. So when you put lime inside the milk, the protein casein denatures, and as a result, it then coagulates. This is a very good example to explain pH denaturing. So I'm sure you also have that experience. You've seen it before, and that it's used a lot in the preparation of cheese. That's how cheese is actually produced. So when I say protein denaturing, it's not necessarily a bad thing. That's what I mean. So if it's in food, it's not necessarily a bad thing. But if it happens in a biological being, in our bodies, that's when it can be a bad thing. A very good example of denaturing, this time with heat, is what happens with your egg. So if you look at this picture, you have your egg white and your egg yolk. The protein in egg is albumin. So think about what you do when you're frying your egg. You're denaturing the protein. And in the process of frying your egg, look at the transformation that happens in the egg, from this form to this form. This is your sunny side up. Some of you like it, Okay, this is what is called the sunny side up. So when you're requesting for sunny side up in a restaurant, this is what they give you. So know what you're asking for. Okay, so what happens as a result of heat? Hydrogen bonds are destroyed, then the globules unfold, the helix, the alpha helix uncoils, and of course biological activity is lost, but this is the transformation that happens in that protein. So it goes from this to that. So we already talked about the pH denaturation in the case of casein, and I told you, for example, that that's how cheese and yogurt are produced. So in the food industry, protein denaturation is actually used to produce different dairy products, and it's also used to enhance and improve the taste of this product. So implications of protein denaturation, especially in food. So the first one is on improved digestibility, and what happens because you're exposing the peptide bonds, and you're making it easy for the enzymes, the digestive enzymes, to hydrolyze those proteins. So this happens during digestion. You've done digestion, and you're aware about the enzymes that are found in the pancreas, the pancreatic digestive enzymes, those in the stomach and pancreas, the proteases. So protein denaturation helps those enzymes get access to the proteins in our diet and to break them into their small peptides or amino acids for absorption. So this is a good thing, and this facilitates bioavailability in the sense that those amino acids can then be absorbed into the body, into systemic circulation, as it were. So they are available from the food, and they are transferred through the gut and the small intestine, and they are absorbed into systemic circulation. And the last one, one of the implications of protein denaturing is that there's reduced anti-nutritional activity. So sometimes our foods have anti-nutrients, as we call them. So for example, in some legumes we have protease inhibitors. So you have some substances in those foods that inhibit certain enzymes. So denaturing, the natural denaturing that happens in the stomach actually can help to denature those proteins inhibitors and prevent the anti-nutritional activity of those foods. So when we look at the biological functions of proteins, it's good to remember, when we think about proteins in a nutshell, to remember, so our enzymes are proteins. So all the enzymes of digestions, the enzymes chymotrypsin, amylase, trypsin, amyloglucosidase, and all the enzymes, the digestive enzymes, don't forget that they're all proteins. When you think about structural functions, your hair, your skin, they are all proteins. When you think about transport, think about hemoglobin. So if you're thinking about the functions of proteins, myoglobin, hemoglobin, they are all proteins. We already mentioned a hormone that is responsible for transport of sugar from the blood into the cells. The hormone insulin is a protein. When you think about your immune system, the antibodies, immunoglobulins, they are all proteins. So when you're thinking generally about protein function, you can think about your enzymes, you can think about your keratin, proteins in your skin, in your hair, you can think about the transport in the blood, you can think about hormones and the numerous hormones, and you can think about our immune system. So this shows us that proteins play a very, very important role in our survival and in our metabolism and in the function of our entire organism. So in this slide, we shall be looking at the quality of proteins, and this is one of the um, health implications of proteins, and we'll be considering the proteins in our diet. And these days we have lots of of, of foods. We have lots of eateries. We're exposed to lots of foods being produced by individuals, a lot of them street foods, a lot of them not much being regulated. And as nutritionists, we often ask questions about what constitutes a high quality protein. So we always refer to, you know, protein quality, and it's important that you also think like this, and to know what protein is high quality, what protein is not so high quality. So we'll be looking at the elements of this, and indeed, I'm sure by now you're beginning to see that it all boils down to the amino acids, the essential amino acids. So it's an important quality of the foods. So you're asking yourself, which foods have the essential amino acids, which foods do not have the essential amino acids. It's good to know this because there are some foods that usually don't have essential amino acids, and there are some foods that do. So your knowledge of this is very important so that you can know how to combine foods in a way that you can have an improved quality, and the quality of your diet can be very much improved. Okay, so we have terminologies that we often refer to. So one of them is high quality proteins. And what are high quality proteins as far as our diet is concerned? High quality proteins are proteins that we call complete proteins because they have all the essential amino acids. Remember them? The PVT team Hall. So PVT team all is what you always have to watch out for in any diet, and you have to ask yourself, this protein that I'm getting from here, from there, does it have all the essential amino acids? So as nutritionists, we have to know this, and a meal or a diet that has all this we call a complete protein in that sense. So still on protein quality, obviously a lot of qualities influence protein quality, and one of those qualities is digestibility. So the fact that your body is able to digest the protein and expose the amino acid. So for the amino acids to be available in the blood, they have to be digested, right, and broken down into the amino acids, and of course those amino acids need to be absorbed. When they are absorbed and they are in the systemic blood circulation, obviously we say they are bioavailable. So digestibility, bioavailability is very important for protein quality because of the reasons I've just explained. Now, animal proteins, like egg, like milk, like meat, are generally considered to be high quality. Why? Because eggs, milk, and meat, they usually have the essential amino acids. However, plant proteins are limiting. We call them limiting because they lack one or more essential amino acids, and that's the reason why sometimes we combine different foods, especially plant-based foods, in our diet, so that where one amino acid is limiting, the other one can compensate, so that we can have a complete protein diet. So a very good example is the reason why you eat rice and beans. So rice is a cereal and beans is a legume. So rice is deficient in some essential amino acids, and beans have those essential amino acids. You need to find out why. So could you find out what amino acids are deficient in rice that are present in beans, and vice versa? But in any case, combining rice and beans, especially for, you know that there are some people that are vegans. They don't take meat products, so they don't take animal products. So this is very, very, very important, so for vegans, they have to carefully use their nutrition to combine different plant products so that they are not malnourished and they are exposed to all the essential amino acids. Another important concept that we often talk about is amino acid balance. And what is amino acid balance? It's used to describe the proportion of essential amino acids that is available for protein synthesis. So the body is constantly making proteins from, the amino acids that is from the proteins that we take in our diet. And when a diet is deficient in some essential amino acids, we say that diet is limiting. Why? Because if you remember that when I was explaining that when the body is synthesizing a protein during protein synthesis, imagine for the protein to be the amino acid combination of that protein is this. So imagine this is the amino acid sequence of that protein. If the body lacks an essential amino acid, the body will not be able to make that protein. So it will truncate automatically. So imagine this is the protein that needs to be produced. Which of this sequence is an essential amino acid? So if you remember your PVT team, so you see methionine or so histidine is an essential amino acid. So what it means is that if the body needs to produce perhaps, let's say, this enzyme, and histidine is required, it means that the whole process will be truncated. It will not be produced because histidine that is needed is unavailable. So we call this the limiting amino acid in that sense. So it's that amino acid that is required for the production of a particular protein, and the balance in the body in terms of availability of that amino acids. Talking about amino acid balance, we'll look at combinations of different diets like we've examined before in the previous slide, where you combine cereals with legumes to improve amino acid balance. So that was the example of rice and beans that I mentioned about earlier on. So this is the nutrition logic behind combining rice with beans. So imagine if you have a situation, either you are a vegan So some people are vegan. They are vegans by choice. This is on a lighter note, while others are vegans by condition. Okay, so whether you're a vegan by choice or you're a vegan by condition, well, let's say you're having your meal and for whatever reason they say there's food insecurity in Africa. I don't know if it's a good example, but maybe you can't. Your protein is expensive, or you choose to be vegan, whatever. Okay, so if you're not gonna have egg in that dish, you're not gonna have meat in that dish. Well, you have to combine your plant protein in a manner that you get all the essential amino acids. So that's what is important here. And what an imbalance will do will be that your body will start breaking down your own proteins. The body will start breaking down your muscle proteins to generate or synthesize that essential amino acid. So that is actually what happens when you don't supply them in the diet, and we call that amino acid oxidation. And during amino acid oxidation, what happens? Amino acids are broken down for energy and converted to other metabolites that are needed for protein synthesis. So this happens when those amino acids are either in excess or they are inadequate. So earlier on in the previous slide, we talked about limiting amino acids. So what are they by definition? I explained them, but if you need a definition, the limiting amino acid is the specific essential amino acid found in the shortest supply in the food relative to what the body needs for protein synthesis. So if you remember that example of a protein that needs to be produced, but that protein is made up of different amino acids, but if those amino acids are not available, the process is truncated. In this slide, we talk about more concepts and we look at complementary proteins. And we've talked about it earlier on. So when you combine rice and beans, what are you doing? Well, you're complementing the protein. So rice is deficient in certain amino acids and certain essential amino acids, whereas beans is sufficient in those amino acids. The same thing, maize and beans. Maize is deficient in certain essential amino acids, whereas beans is sufficient in them. So this process of combining two plant-based foods in which one is deficient in some essential amino acids and the other is sufficient. So the process of combining those two foods so that you have all the essential amino acids in your diet is called protein complementarity in that sense. Okay. So obviously when you have all the essential amino acids in your diet, you can say that, well, that diet has high quality or high protein quality, right? Okay, and as we have said, that foods that have all the 9 amino acids in their right proportion, they are called complete proteins. And we used examples of egg, dairy, and meat to represent complete proteins. So in this slide, we look at the role of proteins in growth. And we all know, we've known for a long time that proteins are very essential for growth. Proteins are needed for cell division, organ development, skeletal growth, especially in children and adolescents. So that's why exposing children and adolescents who have growth spurts at different ages. There are growth spurts where children grow faster than usual. There are different points at the point in which they are adolescents and at the point in which they are children. So it's very important that this protein diets, their diets are rich in proteins, because at that stage they need they undergo rapid cell division, and for a fetus, for example, lots of organs are being developed. For a child, the organs are maturing. So these stages are stages in which you need to expose them to high quality diets, and they're even more important because there's a high turnover for those proteins, and because they're constantly being used for this development, they need to be replaced in diet to replace damaged tissues. And also proteins that are needed for the synthesis of antibodies in the immune system. So it's part of the turnover. Proteins are needed for cytokines development, so they need really to be in the diet. And what happens during inadequacy, we see it evidently as growth impairment, and we see it in a stunted child, in a wasted child, in an underweight child, and we see it in cases in which wounds are not healing fast, and we see it in cases also that the immune systems are weak.
Acute malnutrition prevalence assessed by through MWAC and ODIMA shows that in northern Yobe we have 11.3%, and then central Yobe we have 8.6%, while in southern Yobe shows that it has 6.5%.